Hello and welcome everyone. In this podcast we're going to talk about protein function. We're going to begin by talking about ligands and binding sites on proteins, and then we're going to move into some specific examples of the importance of these proteins' ability to bind substrates. In the last podcast, or the last couple podcasts, we talked about the structure of proteins, the levels of protein folding. We talked about the importance of proteins folding properly. Early on, we talked about some examples of what proteins can do in a cell. We said they could be enzymes, they could provide structure, they could provide storage, they could signal events from one cell to the next cell or within a cell. They can act as receptors, which help control various senses like sight. They can move things within a cell. They can regulate cellular events like transcription and replication. They have a lot of functions. But all proteins can do one thing. They can bind other molecules or themselves. And they bind with specificity. Proteins can bind one, sometimes two, maybe a third thing, with a high degree of specificity. They typically just don't bind anything. There's a degree of binding we should just remind ourselves about. Sometimes they bind their substrate tightly, and sometimes they bind their substrates more transiently. And of course, a range in between them. We talked about this term a few podcasts ago called the Km value. To remind you, the Km value is the concentration of substrate when the enzyme reaches half of the maximum velocity. So if we have reaction rate over here, and on the horizontal axis we have substrate concentration. Many enzymes will do something like this, where Vmax is here, the maximum velocity of the enzyme. Roughly, this is half of Vmax. And so this is our Km, or the Michaelis constant. So how can we interpret a Km value? We mentioned this before, but it bears mention again now that we're talking about protein binding. A small Km means that there is high affinity, meaning it's binding tightly to the substrate. And a larger Km would suggest a lower affinity. A smaller Km, like I've drawn here, indicates that it doesn't require as much of a substrate concentration to reach the half of the maximum velocity. A larger Km just means the affinity is lower for the substrate, so it requires more substrate to reach half of the maximum velocity. Okay, now that we've been talking about the protein's ability to bind molecules and the degree of specificity, let's spend a little bit more time talking about the parts of the protein that bind substrates. We often talk about enzymes in these discussions, but I want you to recognize that this also applies to proteins in general, because even if they're not enzymes, they still bind other molecules. So let's talk about ligands. Okay, now let's begin our discussion of ligands and binding sites. First, let's give a definition of them. A ligand is a molecule that binds to a specific part on a protein. Other terms for ligand that are more specific might be antigen, and this is a term that we might use more specifically with our discussion of antibodies. Antibodies, they bind ligands, but those ligands in this situation we would call an antigen. Sometimes we might call a ligand a substrate. Often when we use the word substrate, we're talking about enzymes. Okay, now let's give an official definition of binding site. Binding sites are usually found on the surface of a protein. Not usually in the middle of the protein, because it's hard to bind something if it's in the middle of the protein. usually, though not always, but usually, this binding site takes a form of a cavity. It doesn't have to be a real deep cavity, but it usually is almost like at the very least, some sort of indentation of the protein. At this site, it interacts with another molecule. That is, it interacts with a ligand. The way the binding site and the ligand interact is through non-covalent interactions. In antibodies, we will call the binding site the antigen binding site. Pretty descriptive, not too surprising. It's a part on the antibody that will bind the antigen. When we're thinking about enzymes, the binding site is termed the active site. So let's draw an enzyme. And then let's draw a substrate. And let's draw another substrate. You could imagine how these two substrates might come in contact with the active site. So let's go ahead and remind ourselves that what the active site is, this whole area here. Then of course remind ourselves that these are substrates. So you can imagine how these substrates might come in contact and may even bounce into this enzyme. When this substrate bounces in here, you can see by the way I drew it that it doesn't fit in nicely. It may enter this active site, but it's probably not going to stay very long because it doesn't fit nicely. And it's not just about fit. It's not just about this enzyme landing in this landing zone and hanging out for a while. That's not what's happening. It's coming in here, and I'm going to draw it again, but this time situated in the active site. And once it's in the active site, that's when the non-covalent bonds become important. So the ones that are particularly important would be, and I'm going to just draw them right here on the substrate, would be electrostatic. So any of these ionic bonds. So you might predict along the active site here would be the side chains with positive or negative charges. So arginine, lysine, histidine for the positively charged amino acids, or glutamic acid and aspartic acid for the negatively charged amino acids. You could also have hydrogen bonds. In that case, you would expect one of various polar molecules to be along here, which could include the positively and negatively charged amino acids, but also any of those other polar amino acids. You could also have our good friends van der Waals, which are slowly becoming my favorite interactions, where you would have on the substrate transiently positively or negatively charged molecules interacting with these polar molecules on the active site. Now, it's not all about polar amino acids. We could have some hydrophobic, in which case these would also need to be non-polar amino acids to match up with these hydrophobic interactions. Now, it would be unusual in this that they would be right here on the outside of the protein, but you could imagine in certain hydrophobic environments, say within a membrane, where proteins might interact and would interact with hydrophobic molecules within a hydrophobic environment. I'm going to erase these two substrates, this correct one which has successfully bound in the enzyme. So let's go ahead and erase that one. And I'm also going to erase this incorrect one because remember, it tried to bind, but the non-covalent interactions here didn't match up well with this substrate. So it didn't stay in the active site. So remember from way back when that non-covalent interactions are weak by themselves, but strong when together with other non-covalent bonds. So you can imagine that the relative affinity for this active site by the substrate is dependent upon the number and the types of non-covalent bonds here. Electrostatic, you might predict, would be stronger than van der Waals. If this interaction is dependent upon van der Waals, then it's probably going to be a very low affinity. A lot of electrostatic interactions, a higher affinity. So I'm going to write here the number and types of non-covalent bonds largely determines, it's not the only factor, largely determines enzyme or protein affinity. I drew this as an enzyme because enzymes offer a nice example of illustrating these interactions. But proteins that are not enzymes also bind molecules, and they also take advantage of these non-covalent interactions. Now let's go ahead and finish this substrate off. Remember, it binds into an active site, not just so it has a place to hide for a while. It binds into this active site so it can be converted into a different form. And it does so in many ways, and we've talked about this before, so I don't want to go into great detail, but sometimes this enzyme may apply the right amount of pressure to the substrate to encourage an event like hydrolysis, so you'd create two new molecules. Or it might bring in two separate substrates and link them together by providing that right arrangement of atoms to cause those bonds to form. For this case, let's go ahead and have it cause some sort of hydrolysis, where it's breaking the substrate into two parts. And by breaking into two different parts, you could imagine how these non-covalent interactions would be disturbed, allowing the two substrates to pop out of the enzyme. So let's go ahead and remove the original substrate through the power of magic here. One thing that I didn't indicate when I drew this is sometimes when the original substrate is in here, it may cause the enzyme to change shape a little bit in order to facilitate the actual catalysis. But once they've been released, the product, the enzyme will bounce back to its original shape. And that's important because that illustrates an important part of enzymes and proteins, is that when reactions like this occur, the enzyme is not consumed. It lives to fight another day. If you had to make a new enzyme for every time you wanted to catalyze this particular reaction, that would not be very efficient. Enzymes are often reversible, where under the right energy requirements, and it may not be energetically favorable, but these two could enter back in here and reform the other substrate. During the process of catalysis, the substrate, between substrate and product here, the substrate will form transient conformations. Not very stable, but it doesn't usually go from substrate to product immediately. There's usually a transient conformation of that substrate. And this is a last thing I want to say about enzymes in this whole process of catalysis, is that they decrease the activation energy, whereby they increase the rate of the reaction. They don't make unfavorable reactions favorable. They just change the rate of reactions. So these are some key characteristics of enzymes. A couple questions I want to answer real quick here that popped in my mind as I was drawing this is, can this active site accept more than one type of substrate? Not that can two substrates of the same substrate bind here, but could it accept a substrate that looked like this, which would be its natural substrate the way we drew it before, but could it also accept something that looks like this? And the answer is yes. And sometimes this might be of biological importance, where you regulate the enzyme this way, or maybe the enzyme may have a couple different functions, but typically an enzyme will bind one substrate to produce one product. Now, where this comes into play more often than not is sometimes there will be substrates, perhaps pharmaceutical substrates, that can bind into these active sites and act as inhibitors. So this is an important concept, and we'll come back to that later, but you'll have to watch the rest of the podcast to see it. How's that for a tease? So these inhibitors will bind here, and then they can inhibit the function of the enzyme. But typically, naturally speaking, there's one enzyme, one substrate. The other question I want to address is, are there more than one binding site? We talked about this binding site, but could there be another binding site on this enzyme? Well, it's not required that it has more than one, and it's not required that it only has one. Sometimes there might be another binding site, say here. It could really be anywhere, I just drew it here. And sometimes these binding sites can enhance a reaction. Maybe it's a cofactor binding site where a cofactor comes in and can bind there. But again, just like we talked over here by answering this first question, sometimes inhibitors can bind there. And again, we'll come back to inhibitors binding either here or here in a little bit. Next, I want to talk a little bit about antibodies. I want to talk about them so we can have another example of the importance of binding sites and substrates. Though we won't go into great detail, if you want to learn more about antibodies and our immune system, you should take one of our immunology classes. So here is an antibody. Kind of a generic looking antibody. I want to point out a few things on this antibody. This region here is the constant region. This constant region determines how the antigen will be destroyed. So another way of thinking of this, it determines what kind of antibody it is. There's antibodies called IgM, IgD, IgE, IgG, and within each of those classes, this will be constant. It'll be the same. But between the different classes of antibodies, it will be different. Now this region here, as well as this region here, we call the variable region. This variable region in itself is not really large. It's between 100 and 130 amino acids. Okay, within this variable region site, this is where we will find our antigen binding site. Antibodies are produced, some of them are produced when we're very young, and then some of them are produced throughout our lives in order to fight different kinds of infections or to destroy disease cells like cancer cells. And because there are potentially, as your book says, billions of foreign antigens, ones that could cause us harm, there's almost a limitless supply of variation within this variable region so that we can fend off the multitude of attacks that we may receive from various kinds of infections that we encounter throughout our lives. So briefly, how does this work? So if you think of, say, our circulatory system, we have a lot of cells floating around that we're going to call self cells. So maybe there's a red blood cell here, a white blood cell. But you have a lot of these self cells, meaning they are our cells. We don't want the antibody to destroy these. But just in the same respect, these antibodies are floating all around. But they know not to attack these. And to understand why they know that, we have to go way back and talk about how they're made. And we don't really have time for that in this class. But let's say a bacterial infection comes in. That's what I'll talk about now, but the same could be true for a virus, a cancer cell, and any other kind of infection. These antibodies will bind here. And in fact, they do it in such a way where different antibodies, because remember, on the outside of this bacterium, there are lots of different antigens, the substrates for the antibodies. Lots of different antigens. And the antibodies are able to recognize various forms on the outside of this cell. This is really helpful because it makes it harder for the bacteria to adapt to this attack because we're attacking the bacteria at multiple sites. Once they've attached to the bacterium, so after these antibodies have coated the bacterium, there are two results that we will talk about. The first is it could cause the bacterium to lyse, which would kill the bacterium. The other is that once coated with these antibodies, it targets the bacterium for destruction by various other cells that can phagocytize it, such as macrophages. But it's all dependent upon, and this is really kind of the key that I'm trying to make here, is that the antigens bind to the variable regions of these antibodies, again, because of non-covalent interactions. It has to bind very tightly here because you want a high degree of affinity so that the antibodies don't fall off. This isn't like an enzyme where we want them to bounce on and bounce off. We want them to bind and attach strongly so that it can either lyse the cell through various mechanisms or target it for destruction by macrophages. Again, it's key, these non-covalent interactions between the antigen binding site and the antigen. Okay, I want to close with this last topic, and I'm going to come back to enzymes as promised. And I want to talk specifically about inhibitors. we have two classes of enzyme inhibitors: competitive inhibitors and non-competitive inhibitors. Let's start with competitive inhibitors. And we mentioned this before, but let's draw it again. We have our enzyme here, and I'm going to draw it like such. And it's supposed to, under physiological conditions, supposed to bind, let's say, this substrate. That's what its job is, to bind this substrate and to make a product. However, sometimes there are other substrates that can compete with this active site. And they look often similar on the site that binds here, but they may have a different conformation on the other site. Now, the only way this becomes a problem for this enzyme is if this has a higher affinity or there's a higher concentration of this enzyme. At some level, this competitive inhibitor has to have a higher chance of binding to this site. And if it's binding to this site preferentially over the natural substrate, then this enzyme won't be able to do its job. And if it's an essential enzyme, say like a glycolytic enzyme, this could be a lethal competitor. So let's talk about these non-competitive inhibitor sites. Well, let's draw a similar enzyme, and we'll draw a similar substrate that binds. But this time we're going to draw, as I did earlier, a different substrate binding site. And let's say when this inhibitor binds, by binding to this site, somehow the active site over here changes. Maybe it changes in such a way that the non-covalent interactions necessary for the binding of the proper substrate are no longer there, or no longer accessible to the sites needed on the substrates. In this way, this inhibitor prevents the substrate from binding, but not by competing for the active site. Sometimes it doesn't necessarily change the ability of the substrate to bind the active site, but once in the active site, the conformational change that occurs in the enzyme won't release the substrate or won't properly create the products so that the products can be released. The substrate's just kind of stuck in there. In thinking about a competitive inhibitor, if the system wanted to prevent the competitive inhibitor from having an effect, more substrate could be added. And with more substrate, it would eventually gain an advantage over the active site. For non-competitive inhibitors, the amount of substrate usually doesn't matter, because as long as this is binding in here, you've changed the active site in such a way that whether or not you have a very small concentration of the inhibitor of the substrate or a large concentration of the substrate doesn't matter, because this little guy here has already messed everything up. Now, pharmaceutical companies work with enzyme inhibitors all the time. They're constantly looking for enzyme inhibitors. Maybe it's because they want to decrease completely the effect of an enzyme, or maybe they just want to slow it down. Maybe there's an enzyme in our system with certain diseases that is functioning at a higher rate than it should. If you could have an inhibitor that competes with that active site, maybe you would be able to slow down this enzyme just enough to resume to average conditions. But these are a very hot topic for pharmaceutical companies. All right, that's all I have to say for now. So let's end with a quick summary. We talked about a few things today, but in essence, it was all centered around protein function. I'll put protein/enzyme. And of course, when I say enzyme in this context, I'm only talking about enzymes that are proteins. We talked about how the active site was important and defined that, or more generally, we should call that the substrate binding site. We talked about how ligands bind to these active sites. And we talked about the importance of non-covalent interactions that help determine the overall affinity of ligand to active site. We then talked about some examples. We talked about very specifically enzymes. And we talked about within enzymes, we talked about inhibitors, competitive and non-competitive. We talked about antibodies and how their variable regions allow for proper non-covalent interactions along the outside of pathogens and target those for destruction. We talked a little bit, not much, but we'll probably hit on this later on, how we use this information. Maybe from the pharmaceutical scientist position, developing new drugs with these inhibitors. From an antibody perspective, certainly pharmaceutical companies would be interested in that, but also we use antibodies in research all the time so that we can identify and label certain proteins. All right, that's all I have. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.